Hi, good morning, everyone, and welcome to the Bio-geotechnics online seminar series organized by the Center for Urban Solutions at NTU. I'm Professor Chu Jian, the director for the center. And today we are very fortunate to have invited Professor Ningjun to deliver today's lecture. Professor Jiang is a leader in the bio-geotechnics research, and he is a very accomplished researcher in geotechnical and geo-environmental engineering. Professor Jiang received his PhD from University of Cambridge on the bio-geotechnics topic. And then after that, he joined University of Hawaii in the US for a number of years as a faculty. And before he moved to Southeast University in China. By the way, and Southeast University is one of the top universities, particularly in civil and transportation engineering. And so Professor Jiang has received a lot of awards, including the Fairland Awards in 2019 and from Canadian and Geotechnical Society. And he is also one of the recipients for the Best Paper Awards in 2020 and from one of the high impact journal, ECTA and Geotechnica. And he's also the recipient for the Science Foundation, which is another top geotechnical journal, the editorial board member award. And recently, he is a recipient of the Geotechnique, which is one of the top geotechnical journal, again, the Early Career Award just recently. And Professor Jiang is also an associate editor for a newly established journal and bio-geotechnics. And he is the editorial board for a number of journals, including environmental and geotechnics. And next page, yeah. We are, this bio-geotechnics is one of the most hot topics in geotechnical engineering. And so for that reason, we are going to organize the next conference on microbial biotechnology in construction materials and geotechnical engineering. So it deals with not just geotechnical, but also construction materials as well. When we say bio, it does not limited to bacteria. It can also be the use of vegetation, or anything to do with a bio process, including the use of polymers and use of other additives to innovatively improve the construction materials as well as the geo materials. So if any of you is interested, you can get online information and submit a paper. And the conference will be from 15 to 16 August next year. So without further ado, and now let's welcome Professor Jiang to deliver today's lecture. Yeah.
Thank you. Thanks, Professor Chu. So I will share my screen. Okay, so good morning, everyone. I'm Ningjun Jiang from Southeast University in China. So today, I will share the research of my research team about the microbial mineralization in soil, in particular, the applications in geotechnical and geoenvironmental engineering. So firstly, I would like to introduce my team members. So the first line shows my current members, mostly are PhD and master students. And the second line shows the previous members, they were PhD students as well as some research assistants. So without their hard work, it's impossible to get this work done in recent years. And I would also like to thank these funding agencies like the State of Hawaii Department of Transportation, the National Nature Science Foundation of China for their support in my research. With their money, it's only possible to do good research. Okay, so for today's presentation, firstly, I would like to briefly introduce the background of biosoil research. Then I will spend most time introducing three studies done by our research team in recent years. One is for soil hydraulic erosion control, second is for calcareous sand stabilization, and third is for heavy metal immobilization in sandy soil. Then finally, I will take like a few minutes to talk about the future for MICP. Okay, so as we know, in classical soil mechanics, we normally regard soil as a three-phase substance consisting of solid, water, and air. And all modern soil mechanics theories are based on the three-phase framework. So it means that we only consider the inorganic matter in soil. We ignore all biological activities and substances in soil. So from the perspective of like agriculture and environmental science and engineering, it's not correct because biological activity plays a very important role in soil. It can change soil chemical and biological properties, can affect a lot of things. But in soil mechanics, we simply ignore it. So in recent years, people gradually recognize the importance of biological things like plants, like bacteria in soil that could affect the soil engineering properties. So that leads to the establishment of bio-geotechnics, a very new branch of geotechnical research. Okay, so in bio-geotechnics, currently people are focusing on several processes like biomineralization, biogas, or biofilm. Then among these processes, the biomineralization, or more specifically, the microbial mineralization, is one of the most popular area for research. So actually, the microbial mineralization is a quite ubiquitous process in nature. It can be driven by many processes, including the oxidation reduction. For example, the reduction of sulfate is a quite common process, like the photosynthesis, because the plants can absorb the CO2 from the air to produce carbonate, and this could lead to mineralization. And among these microbial mineralization processes, the microbially induced carbonate precipitation, or MICP, is a key research area in geotechnical engineering. So here, when we talk about MICP, we normally refer to the urealytic-based MICP. So since I believe the audience today are quite familiar with MICP, so I basically just briefly introduce the process. So for urealytic MICP, so in this process, firstly, some urease-producing bacteria, it could be standard bacteria or it could be native bacteria, could secrete urease. This is a special enzyme. This enzyme could hydrolyze urea to produce ammonium and carbonate. And meanwhile, the environment pH also increases, so it becomes alkaline. Okay, so under this condition, if there's also calcium ions present in the system, then the calcium ions could react with carbonate ions to generate calcium carbonate. And this leads to the crystallization and mineralization. Okay, so the formed crystals, if happening in the sandy soil or granular materials, it could cement the granular soil particles. So this could lead to the improvement in soil engineering behavior. So previous researchers about MICP have revealed that the mechanisms of MICP for granular soil improvement mainly on these 3 points. First one, the generated calcium carbonate crystals could provide bonding strength. So it means that the cohesion could increase. And secondly, because we have extra crystals within the soil, so it means that the overall density of the soil is improved. Improved density normally means improved strength. And thirdly, some formed calcium carbonate precipitation could also coat the soil particles. So it could change the particle roughness, normally increase it. So this can also give additional contribution to the soil strength. So considering these 3 mechanisms, then MICP is quite promising for granular soil stabilization. Then next, I would like to talk about how we can implement MICP potentially in the field. So from the perspective of environmental engineers, there's 2 ways to implement bio treatment. The first one is bio-augmentation. The second one is biostimulation. For bio-augmentation, it means that some standard known bacteria which are cultivated in the laboratory directly injected in the form of solution into the ground, followed by the cementation solution injection. And this could lead to the MICP reaction in the field. Of course, this one is quite convenient because we simply need to prepare existing bacterial solution, then do it. But the disadvantage is that the compatibility of the injected bacteria with the ground, with the in situ soil may be a problem. They may diminish very quickly. So that's bio-augmentation. Then the second approach is biostimulation. So actually, there's also 2 ways to do biostimulation. The first one is ex situ biostimulation. It means that the native soil is firstly taken back to the lab, and then extraction, isolation, and purification of native microbial species are performed so that we can get adventurous indigenous bacteria. This one could work very well in the native soil condition. Then we could perform enrichment and reinjection to get the bacterial solution back to the ground. Of course, this one makes the compatibility much better than the bio-augmentation approach. But as you can see, it could take a lot of effort to do extraction, isolation, purification, and enrichment. And potentially the associated costs are high. Then the other way to do biostimulation is the in situ biostimulation. It means that instead of taking soil back to the lab and do a lot of analysis, we simply inject enrichment medium directly into the ground. Here we need to carefully design the ingredients of enrichment, making sure that it could work effectively to stimulate indigenous bacteria. And in this approach, it is not necessary to know what exactly bacterial species are stimulated. Instead, we only need to control like the ureatic activity sort of things so that we know it works. Then it could do MICP. So that's the 3 approaches that can be used for MICP implementation in the field. And regarding previous MICP research at different scales, we can see a lot of reports and technical papers. So from nano to micro scales, people have used like SEM to get the microstructure features, use atomic force microscope to see the interactions between particle and the bacterial cells, use the microfluidic chips to see how bacteria and cementation solution migrate within the porous medium, to use XRD to see how crystal changes with time with the ingredients. At the element scale, researchers are using various geotechnical testing methods. For example, the traxel shell test, unconfined compressive stress test, compression test, etc. So to assess the mechanical behavior of MICP treated soil. And at pilot to field scale, we can also see a lot of reports. And this is an interesting part, so I want to introduce a little bit more. So here, this slide shows, some representative pilot to field scale applications of MICP in the last decade. For example, like in 2013, Professor Dion from UC Davis used MICP to do surface erosion control. Like Professor Chu in 2013 also used MICP to make a pond, to make it impermeable. Professor Liu from Chongqing University used MICP to treat calcareous sand in South China Sea, and they got the solidified, sand foundations. Also in 2019, Professor Van Passen did the liquefaction control using the MIC method, and we can see the setup in the field. In this year, Professor Kawasaki from Hokkaido University in Japan used the MICP method to improve the slope stability, and this graph shows the laboratory model test. And in 2021, Professor Kawazanjun from ASU treated the sandy soil in the MICP, and we can see the formed column with quite high strength. Professor Miao, also from Southeast University, considered the wind erosion in the desert area using MICP and achieved a quite good result. Professor Montaya from North Carolina State University used MICP for coastal erosion control, and this is the large-scale model test. And also some other researchers use MICP for contaminant immobilization and control, and here is the setup for the monitoring. So we can see there's already many researchers trying to upscale MICP to the field. So that's good. But to be honest, there are still some problems that as a bio-geotechnics community members, we have to address them before it can be used in a real project. And here I listed 3 papers our team published in the last 4 years. So they are mostly review papers talking about the future of MICP for soil improvement, a comprehensive review of the process, materials, calculation, and applications of biomedical soil improvement. And for the paper published in bio-geotechnics this year, we reviewed the MICP method in environmental geotechnics in particular. So if you are interested in these papers, you can simply search online and find them. Okay, so second part, I would like to introduce our research on MICP for soil hydraulic erosion control. So as we all know, the hydraulic erosion problem could lead to disastrous engineering problems like the dam internal erosion, like the seepage piping, coastal erosion, rainfall erosion. They happened globally, almost everywhere, every year. So it's very important to find a way to address it. So here we propose the use of MICP to control hydraulic erosion. So in order to do that, we have to bear in mind a few key points. The first one is that the hydraulic erosion vulnerability of soil fundamentally depends on soil behavior or soil properties. For instance, the particle size distribution, the fines content. So our target should be erosion vulnerable soil. It means that naturally the fine particles from the soil can be eroded. Okay, so that we need to do improvement. And secondly, when we do this research, we need to consider the coupling effect because there is a quite complex mechanical, hydraulic, chemical, and biological coupling when we use the MICP to control hydraulic erosion. Now, how to describe this process is very important. And thirdly, we need to think about how to assess the erosion control effectiveness using MICP. So that's the three key points we bear in mind when we did this research. Okay, firstly, this is an example we did for the internal erosion control in a grass sand mixtures. Because in grass sand mixtures, especially when they satisfy a certain particle size distribution, then the sand particles can be very easily washed out from the gravel skeleton. So firstly, we look at this case. So we designed a large one-dimensional column internal erosion testing apparatus, as you can see here, to accommodate the size of gravel particles. We made it with a diameter of 240 millimeter and height of 550 millimeter. So it's large enough to accommodate the gravel sand mixtures. Then we designed an MICP implementation unit. So the bacterial solution, cementation solution, and distilled water can be injected in sequence based on our need. In addition, we also installed a hydraulic pressure unit so we can achieve different hydraulic pressure levels. And also this sample is controlled on a pneumatic cylinder so that we can achieve different vertical stress levels. Then in this test, we do both untreated soils as well as MICP treated soils under different hydraulic pressures. So this graph is quite obvious. So when the hydraulic pressure is not quite high, we can see only a little bit erosion along the surface for the untreated samples. For MICP samples, no erosion at all. Then when the hydraulic pressure increases to 20 and 30 kPa, we can see very significant surface washout zones. So it means that the erosion happens a lot. But for MICP treated samples, still no erosion at all. So from the visual observation, we already see MICP work very well. Then we want to know what are the key factors that affect the erosion control effect of MICP for gravel sand mixtures. So firstly, we've looked at the cementation solution concentration. And from this graph, this is the erosion rate versus time graph. So from this graph, you can see that the black one is the untreated samples. The green one is the low cementation case. The pink one is moderate cementation case. Then the red one is the heavy cementation case. Obviously, if we only have a little bit cementation, the erosion control effectiveness is not that good. In some cases, it could be even worse than the untreated case. But if we have a lot of MICP, so with two more cementation solution concentration, then almost no erosion. And in addition to the erosion rate, if we look at the erosion rate versus flow velocity relationship, we also find that with heavy cementation, the critical flow velocity is also larger than the moderate or slight cementation case. So it means that MICP treatment could improve the critical flow velocity. And the more cementation solution concentration we have, the higher critical flow velocity we have. In addition to the cementation solution concentration, the original soil composition is also a quite key factor. So here we investigate two cases. One is with 25% sand, the other is with 50% sand. For 25% sand, this is basically a gravel supported structure. It means the skeleton is mostly gravel and the sand particles are loosely distributed within pores. For the 50% sand content case, so this is basically a sand supported structure. It means that the skeleton is formed by sand instead of gravel. So for these two structures, after we did the erosion test, we find that MICP work very well for the gravel supported structures. It can effectively cement the sand particles so that they will not migrate through the pores. But for the sand supported structures, MICP is not always effective. In some cases, it could be worse, especially if we have high level of vertical stress. This is because we simply have too many sand particles. And if the cementation is not enough and we have high vertical stress, then stress can push the uncemented sand out of the soil structure. So we see a lot of erosion mass. And if we see permeability, in this case, so the permeability also increases dramatically because of the loss of sand particles. So this result demonstrates the importance of original soil composition. So in addition to the graph sand mixtures, we also investigated the internal erosion control of sand clay mixtures, because this also happens quite frequently in the field. Because sand and clay particles are smaller than the gravel particles, so this time we made a smaller testing apparatus with 50 millimeter diameter and 100 millimeter in height. So in this test, because we have to monitor clay, and it's almost impossible to measure the eroded mass. So instead of use a balance, we use the turbidity meter to monitor the clay concentration from the outlet, so we know what is the mass of how many clay particles have been washed out. And in this test, we use the peak erosion rate versus shear stress relationship to see how MICP treatment could improve the erosion control. So from this graph, we can see that the blue line represents untreated soil, and the red line shows the pink one and the black one, as well as the red one, shows the MICP treated case. Firstly, we can see that the critical shear stress, which is a turning point between the flat part and the inclined part. So the critical shear stress for MICP treated case is obviously larger than the untreated case. Now, secondly, for the post-erosion part, right, for this part, for the inclined part, so with the same shear stress incremental, we will see much higher increased erosion rate for untreated case than the MICP treated case. So both critical stress as well as the erosion coefficient, the alpha here, the slope, could demonstrate the effectiveness of MICP for internal erosion control in sand clay mixtures. Okay, so in addition to internal erosion control, we also looked at like the sand dune erosion under different wave conditions. Here is a very simple model test in the laboratory. We made sand dune models at different angles from 22 degree to 30 degree, and we made different. We made waves with different magnitude, small waves, large waves, and from this test we can see that in this test actually we used the erosion volume to see how MICP could help to mitigate erosion. And from the test results for both small wave and large wave case, the MICP treatment could reduce the erosion volume compared with the red one. The red one is the control case, untreated case. And actually in this case, we also used EICP to do the treatment. And we found MICP could be even better than EICP in all cases. And the same apparatus, we also use it to simulate rainfall erosion by using some misting nozzle on top, so we can simulate the rainfall at different magnitude. So from this test, we use a photogrammetric approach, the structure from motion, to quantify the erosion. So from the graph on the right-hand side, we can see that the blue part shows the erosion. If we say the elevation change 0 means no erosion, then the minus means that erosion. So for the untreated case, we can see a lot of erosion, mostly uniformly distributed in the center part. For the MICP treated sample, so it's quite different. We almost see no erosion, only a little bit on the lower part, and we could also see some accumulation on the toe part of the slope. So this research could also demonstrate the effectiveness of MICP to prevent the surface erosion of sandy slope on the rainfall. Okay, so that's the first part. The second part, I would like to introduce our research on MICP for calcareous sand stabilization. So since I was working in University of Hawaii for several years, so in the island of Hawaii, the calcareous sand is a big problem because it's vulnerable to liquefaction, and in beach areas, mostly it's calcareous sand. So when we want to build anything on top of it, we have to consider the mechanical property of calcareous sand. So that's why when I worked there, I tried to use MICP to treat calcareous sand. So in this particular research, so we used the biostimulation approach because we think it's more eco-friendly. Then we firstly prepared the enrichment medium consisting of either ether extract or nutrient brass plus some urea and other materials. Then we prepared the cementation solution consisting of calcium, urea, and some trace nutrient additive. Then firstly, it was 48-hour enrichment through surface percolation, so we can complete the stimulation process. Then we took some soil samples and extracted the solution, the pore solution, for analysis such as ureatic activity, pH value, urease concentration, and genetic analysis. Then for the rest samples, after the stimulation process, the cementation solution was applied through percolation once a day for 5 to 10 days to make the solidified soil samples. Then the soil sample was subject to cementation concentration measurement, the microstructure observation, direct shell test, track shell test, odometer test to see its mechanical performance after MICP treatment. Okay, so firstly, let's see after the stimulation process, how bacterial population changes. So for the first column on the left-hand side, that's the original soil without MICP treatment. We can see the existence of many different species, and we don't see some dominant species, and we also don't see some representative ureatic species. But with MICP treatment, either through ether extract or nutrient brass, we could see the dramatic change in the microbial population distribution. So in particular, we could see the proportions of ureatic bacterial species such as Bacillus sporosasana extra increased substantially. So like the red one here, that's the sporosasana. So we can see the dramatic change. So it means that after the biostimulation treatment, we did achieve the purpose of getting more ureatic bacteria in soil. Then after the cementation process, we used a microscope to see how cementation form and how it distributed within the soil structure. So as you can see here, the first graph shows untreated sand, so we cannot see any cementation between soil particles. The second graph shows the slight cementation case, only with 1.86% cementation. So we can see a little bit of cementation between particles, but not too much. Then for next case, it's a heavily cemented case. So the cementation content is more than 6%. So here we can see quite a lot of cementing agent between the soil particles. In addition to the interparticle connections, we could also see some formed crystals coating on the surface of sand particles. Then through the XRD analysis, we could see that after MICP treatment, we have more calcite. So this, for example, this peak and this peak are new peaks compared with original soil. So this could also demonstrate the formation of new calcite precipitation from MICP. Then let's move to the mechanical testing part. So firstly, we performed a direct shell test and created the peak stress ratio with cementation content under different normal stresses. Here, the results are quite interesting. So on the low normal stress condition, so basically the peak stress ratio increased a lot with cementation content. So it means that MICP works quite well. But on the high normal stress levels, like 400, 600, the peak stress ratio still increases with cementation content, but only a little bit. So it means that on the high normal stress conditions or high confinement conditions, although MICP could still work, but it could not help too much because at this time, the confinement dominates the strength of sandy soil. Then from direct shell test, we also obtained the cohesion and correlated it with the cementation content. So we could also see an increasing trend. Then we compared with other published results, like from Cui and Chen. Both of them used the bio-augmentation to the MICP. So compared with their research, we find the cohesion we obtained are larger than their cases. So this is just a premature result to show that potentially biostimulation could work better than bio-augmentation. Then the compressibility, the one-dimensional compressibility test also shows a similar trend. So with increased cementation content, we can see the compression curve shifts upward. So it means that the soil's ability to release compression has been improved. Then we calculate the coefficient of compressibility at different stress range. And similar to the direct shell test results, with the increase in cementation content, the coefficient of compressibility AV drops. So it means that the MICP helps to reduce compression. And then, at high normal stress conditions, then this improvement by MICP becomes diminishing. And I think the mechanism is similar as I explained in the direct shell test. In order to better understand the mechanical performance of MICP treated calcareous sand, we performed both CD and CU traxel shell test. Firstly, for the CD traxel shell test, the first graph shows the post-failure state of the sample under low and high confining stress condition. Second one shows the deviator stress versus effective mean stress relationship. Because it's a CD test, so they are simply the straight lines. And as we can see for the bio-cemented case, the solid line compared with the dotted line, the solid line goes more, right? It means that the strength of the soil has been improved. Then based on the testing results, we obtained the failure envelope both at peak state and at the residual state. And from the failure envelope, we could obtain the effective cohesion as well as effective friction angle. So at peak state, we can see that with the increase in the cementation content, then the cohesion increases. So at 9% cementation, the cohesion could reach more than 40 kPa. Then for the effective friction angle basically is largely independent of cementation level. For the residual state, so firstly, we find that the cohesion almost disappeared. It means that after shearing process, the effective cementation has been destroyed. Then for friction angle, it also does not change too much, but increase a little bit compared with uncemented samples. So here we guess maybe it's due to we have like more cementation or more crystals within the soil structure, even if they are not effective cementing agents, but it could change the soil density as well as the surface condition. So this led to a little bit increase in the friction angle. For the CU test, so first graph also shows the failure state of the samples at 50 and 400 kPa confining pressure levels. Then second graph shows the deviator stress versus effective mean stress. Because it's a CU test, then we can see the change in deviator stress in this way. And the dotted line shows the uncemented samples, and solid line shows the bio-cemented samples. Obviously, compared with uncemented samples, the data, the lines for bio-cemented samples shift to the right. So it means it becomes more like a dense sand. And also by measuring the change in the soil volume, we also find that the bio-cemented sample shows stronger dilatancy compared with untreated samples. Then from the deviator stress versus actual strain curve, we find that firstly, it exhibits the strain hardening trend. Then secondly, with increased cementation level, here the solid line shows the 4% cementation, and the dotted line shows 9% cementation. So compared with the solid line, the dotted line definitely is higher, right? It's higher. So it means that the higher cementation and the confined pressure leads to increased deviator stress. Okay, so then we think about a little more. So we already know MICP could improve the mechanical performance of calcareous sand, but what we can do to further increase it, as well as, and at the same time, addressing the ammonium problem, because as we know, ammonium is a byproduct of MICP. So considering this, then my student proposed to use biochar to do enhancement. So in this particular test, we used two types of biochar. One is like a needle shaped, so quite long and big. The other is in powder shape, so it's quite fine. Okay, so when we mix the biochar with calcareous sand and do MICP treatment, then we measure the cementation content. So first graph shows, a different biochar content from 0.1, 0.3 to 0.5. Then compared with the no biochar case, the cementation content increases. And this is true for different treatment schemes and different initial soil densities. So this result shows that biochar could improve cementation content. So this is very important. Then secondly, we also did the traxel shell test for the biochar enhanced MICP treated soil. And here you can see several lines. The blue line shows the MICP treated samples with the powder shaped biochar, and the green line shows the MICP treated samples with the needle shaped biochar. Then the red one is the MICP treated samples without biochar. If we can compare these three, of course, with biochar, then we have a failure envelope on the top. And from it, we could get cohesion. And if we compare cohesion for these three cases, no biochar, the biochar with needle-shaped biochar and with powder-shaped biochar, then definitely the powder-shaped biochar leads to more cementation and higher cohesion. Then it's the needle-shaped biochar. And finally, the smallest one is the biocemented sample without biochar. So these results could demonstrate how biochar can help increase the strength of calcareous sand. Now, in addition to the strength improvement, the biochar could also help to absorb some ammonium. So this graph shows that during the several cementation solution treatment procedure, we monitor the ammonium concentration within the effluent. And the blue one shows the sample with powder-shaped biochar. The red one shows the sample with needle-shaped biochar. And the black one shows the sample without biochar. So obviously, with the powder-shaped biochar, then the concentration of ammonium decreases more quickly than the untreated one. So these results could demonstrate that the biochar, especially the powder-shaped biochar, could help to absorb some ammonium. So in the future, maybe we could explore more to see how biochar could work effectively in the field application to help to reduce ammonium problem. Okay, then finally, we also took some microstructure photos to see how why biochar could help to improve the soil strength. So the first one is uncemented sand. The second one is cemented sand without biochar. And third one is with 0.5% biochar. So basically for this one, we can see a lot of small dots, right? They are powder-shaped biochar. And we believe these small dots serve as extra nucleation sites for calcite precipitation. So that's why we see more cementation formed in the biochar case. In addition, because we have biochar inside the soil, so it means that the overall density of the soil is also improved. So this is another contributing effect, especially for the post-filial state when the cementation has been destroyed, right? The improved density could help to improve the residual strength a little bit. However, we still need to be careful. The strength of biochar itself is quite low compared with calcite or sand. So it means that it is potentially weak point. So when the soil is very dense, or if we have a lot of biochar introduced in the system, it could potentially destroy or damage the overall strength of the soil. So it means that we have to control the dosage of biochar in soil. Okay, so then based on all these mechanical testing results, we proposed a strength model based on Borton's model for sand. From the Borton's theory on sand strength, we know that shell strength consists of friction, dilatancy, particle breakage, and rearrangement. And the first three black lines shows the original Borton theory. Then, if we need to consider the contribution of MICP for strength based on our direction shell and the traxel shell test results, we think that it helps most at lower effective normal stress or confining pressure range. At this range, we could see the dramatic increase in the cohesion. Okay, so this is the contribution from MICP. However, when the normal stress is high or the confining pressure is high, then the contribution from MICP is diminished. So it becomes aligned with the original filial envelope. Then if we further consider the contribution of biochar, so this is the green line. Firstly, at low effective normal stress range, biochar could lead to more cementation. So we see more increase in cohesion, as you can see here. And at high normal stress level, although the cementation has been destroyed, but the existence of biochar in soil structure, as I mentioned, could change the soil surface morphology, could improve the density. So it could still be a little bit higher than the original failure envelope. So this is the stress model we proposed for both MICP treated case as well as the biochar enhancement case. Okay, then thirdly, I would like to spend some time to introducing the third part of our research, MICP for heavy metal immobilization in sandy soil. So here, in order to understand the process, to optimize the process, we need to keep in mind that the heavy metal species, as well as their concentrations, play a very important role. It means that MICP may only work at a certain range of heavy metal concentrations, not for all. And secondly, it's very important to understand the bacterial activity response, including its population change, its activity change. Thirdly, we need to understand the immobilization process, the crystallization process. Then, in order to understand the immobilization mechanism, we tested the strength and toxic leaching. We tested the contaminant phase change, as well as we established an immobilization model. Okay, so firstly, I would like to talk about how we prepare the samples. So here we use 90% river sand plus 10% kaolin clay to make the sample. Then it is mixed with a certain amount of the lead contaminants. Here we use lead as a representative heavy metal to do the research. Then when we mix it, then we use enrichment and cementation solution to do the stimulation. Here we simply use the premix method. It means that we can only do one-time treatment. After that, the sample cured for a certain period of time before they were subject to like stress, toxic leaching, permeability test, as well as like CFU, urea concentration measurement. So firstly, it's a result about the bacterial and urealytic activity change with time during the biostimulation process. For the first graph, we can see that for the no contaminant case, the black one, the bacteria grows very quickly, and after 4 days, it reaches the peak, more than 10.8 CFU per gram of soil. Then with a little bit contaminant from 20 millimole to 30, then it could still grow very quickly, but there's a little bit delay in the first day, for the first day. However, with even higher contaminant from 40 to 50, we can see the growth has been inhibited, and the peak bacterial population could only reach less than 10.7. Then for the most severe case with 100 millimole lead, it could not grow at all. So the bacterial population is always below 10.4. So it means that in this case, it has been severely inhibited. Then if we look at the urea concentration change with time, because this one could give us an idea how fast the urea has been decomposed. Okay, for the no contaminant case, it is decomposed very quickly. After 4 days, almost no erosion left, no urea left. So it means that stimulation works very well in this case. Then even with 20 millimole, a little bit contaminant, we can see the urea decomposition has been retarded, even a little bit. Then with the increase in the contaminant concentration, the decomposition rate is it becomes smaller and smaller. But interestingly, even if on the 100 millimole case, the most severe case, we could say that the urea could still decompose, although very quickly, but it keeps going. It means that if we left it for sufficient long time, the urealytic reaction could still happen, although in a slow rate. Therefore, ammonia concentration is similar to urea for the no contaminant case. So I see a lot of ammonium. Then with the increase in contaminant concentration, the ammonia concentration becomes smaller. Okay, so this gives us an idea of how bacteria and urealytic activity changes during the stimulation under different contaminant concentrations. Then secondly, is strength and permeability. Although for soil heavy metal immobilization, strength and permeability are not that important, but here we simply look at it, how it changes with time and with the contaminant concentration. So here, interestingly, first we find that the strength increases with curing time. Although the overall strength is not that high, the highest is only like 35 kPa. This is because we only do one-time treatment. Our target is not to achieve high strength, but immobilization is more important. So we don't do high strength, right? Then in this case, we find that after 14 days of treatment, and when the lead concentration is 40 millimole, we have the highest strength. Then for permeability coefficient, we find the same thing. The lowest permeability coefficient happens when the lead concentration is 40 millimole. So that's very interesting. It's not 0, but it's 40. Then we measure the precipitation content. We find that for the 40 millimole case, we have the most precipitation content, more than 0.4%. Not too much, but we have the most here. So this is because we assume that while 40 millimole contaminant could retard the growth of bacteria, but I mentioned the urea decomposition keeps happening. So this reaction keeps happening, MICP keeps happening. So after 14 days, for the 40 case, although we didn't measure it, we can assume still a lot of urea has been decomposed. So the MICP reaction still happens a lot in this case. And because here we have more lead, right? Both lead and calcium can react with carbonate from the urea decomposition to form the precipitation. So that's why we have more precipitation at 40. Then for 50 and 100 case, although we have even more lead ions, but the reaction is simply too slow. So it cannot generate sufficient amount of precipitation compared with the 40 millimeter case. So that's why we think, that's why the 40 millimeter case have the highest precipitation content. Then for the toxic leaching properties, we tested under 3 different conditions: the neutral condition, the weakly acidic condition, and the strongly acidic condition. Because we know the calcium carbonate precipitation is very vulnerable to acid. So normally we don't expect it works under acidic condition. Then from this test, we see similar phenomena. For the neutral condition, after 14 days of treatment, except the 100 millimole, the most severe case, for all other cases, it is below a leaching concentration of 0.1 milligram per liter. So this is a Chinese standard for the type 4 groundwater. So all cases could almost meet it. And for weakly acidic condition, also, except for the 100 millimole case, for the rest cases, 14 days are sufficient. to meet the leaching standard. But unfortunately, for the strongly acidic condition, we can see the leaching concentration is very high. It's all more than 10 milligram per liter, more than 0.1 milligram per liter standard. And as I explained, right, because MICP could not work very well under acidic condition, it could be the precipitation could be dissolved. So it means that if we want to use MICP for heavy metal immobilization, we need to be careful about the acid condition we could potentially encounter in the field. Okay, so then we tested the heavy metal phase distribution. So actually in soil, there are 5 phases. From the stable to unstable, it's a residue, organic bond, the ferromanganese oxide, carbonate bond, and exchangeable. The exchangeable one is the most unstable one. If most heavy metals are in this state, it means that they can easily diffuse within the ground, within groundwater. So after 1 day treatment, we can see that in all cases, most heavy metals are still in exchangeable state. It means that MICP has not yet worked quite effectively here. Then after 7 days of treatment, for low lead concentration cases, so most heavy metal have been converted into carbonic bond. So it becomes more stable than the exchangeable case. For the 50 and 100 millimole cases, still a lot lead in the exchangeable state. Then after 14 days, for 20, 30, 40, and 50 millimole lead concentration cases, we could say that exchangeable state is reduced to less than 1%, and mostly they are in carbonate bond. Even if for the 100 millimole case, the exchangeable state only has less than 20%. So this one could further demonstrate that MICP could help to immobilize heavy metal, could change it from unstable state to more stable state. Then the XRD analysis could help us to reveal what are present in the soil after MICP treatment. So as we can see, for this case, with 40 millimole lead, we can see both calcium carbonate and lead carbonate. We can see both of them. However, for even higher lead concentration case, I didn't put it here, but for that case, we could not find any calcium and lead carbonate. It means that the reaction has been retarded. Then combined with the SCM results, we can further understand it. So in this one, the first graph shows no contaminant case. We can see calcium carbonate, and from the EDS scanning graph, we could also spot this various bright points. They represent crystals in soil. Then for low lead concentration case, we could see both calcium carbonate and the lead carbonate. And from the SCM graphs, we could see the bright spots in this graph and this graph. Both of them represent the crystallization phases. However, for the 100 millimole case, firstly, we couldn't spot these crystals, both calcium carbonate and lead carbonate. Then from the EDS scanning results, we can see that we don't have so many bright spots compared with previous cases. It means that we don't have a lot of crystals in these cases, and this can demonstrate the retardation effect of high lead concentration on MICP. Okay, then we also analyzed the microbial community distribution. So for the control case, we don't see dominant species. Then for the case with MICP treatment but no contaminants, we can see the spore Sassana dominates. Then for the low contaminant concentration case, we can see the amount of spore Sassana becomes smaller, but we see this one, right? This one is become dominant. So this one, after we analyze it, we find it's a lead resistant species. So that's why when we have lead, then it becomes dominant. However, if the lead concentration becomes even higher, then this part becomes smaller. Then, in contrast, the portion for the spore Sassana becomes larger again. So we guess that where we have a lot of lead contaminants, then this one is not as resistant as spore Sassana. So that's why the spore Sassana becomes dominant again under this condition. Okay, then we further performed a chemical equilibrium simulation using MinTech, the software MinTech, to simulate the MICP immobilization process. So as you can see here, so the horizontal axis shows the amount of urease hydrolyzed, and the vertical axis shows pH, quantity of different precipitation species. Then when the lead concentration is 20 millimole, we can see the pH increase very quickly with the amount of hydrolyzed urea. And initially, we can see the lead precipitation happens, followed by the calcium precipitation. Then, if we have more lead concentration in the system, firstly, the increase in pH is delayed. And secondly, we have more lead precipitation at the first stage, followed by the calcium. And if we have even more lead concentration in the system, then the precipitation is dominated by the lead, and we only have a little bit calcium carbonate in the system. And the second row shows different lead precipitation species. And due to time limitation, I will not introduce them in details. Okay, so then based on all this analysis, including the simulation results, we proposed a multi-layer coating model for MICP immobilized lead contaminant. So in this model, so in the center, this is the bacterial cell. Then because as we can see from the simulation results, the lead always precipitates before the calcium, right? So we have the lead precipitates in the center because they are firstly formed, followed by different forms. Then finally followed by the calcium carbonate precipitation. So this multiple layer system has certain advantages because the outer layer calcium carbonate could protect the lead concentration in the inner part. So where, for example, when it encounters with acidic agent, then the calcium carbonate part will react first instead of the lead part. So the contaminant could still be immobilized within the structure. So this helps. Okay, so here are a list of papers we published in recent years covering all three studies I just mentioned. So if you want to know more details about our research, you can check these papers. Okay, so finally, I would like to spend a few more minutes to talk about the future of MICP. So if we want to advance MICP in the future for large-scale field application and has certain commercial values, we need to consider these three aspects. Firstly, it's fundamental. We need to understand better about its fundamentals. Secondly, we need to consider its multiple functionality. And thirdly, we also need to consider the sustainability of this process. So firstly, for fundamentals, we still need to know more about the microbial processes. For example, what is the change in the bacteria from a perspective of molecular levels? How about the evolution of its energy, the energy cycles during the MICP process? That's something we not yet explore a lot. Also, like the chemical processes, how about the hydrodynamic or the kinetics of the crystallization process, and how crystals evolve with time, change with environment? That's something we need to look at. And thirdly, the biochemical fluid in porous media. How we can use advanced model to describe this reactive process. Then for multiple functionality, so while most of current MICP research focuses on soil strengthening, can we also think about how to couple it with ecological, environmental, and carbon footprint benefits? For example, it can both improve soil strength as well as absorb, for example, the carbon dioxide from the air, right? If it can achieve multi-functionality, then its potential use could be quite wide. Then certainly, we also need to rethink the sustainability of the whole process. As we know, right, MICP, during this process, we have ammonium. This is the byproduct. So how we can further reduce the amount of ammonium, prevent it from diffusion into the air. That's something we need to think about. Also, like the ecological balance and restoration. So when we implement MICP in the field, will it change the native bacterial population distribution? So after we complete the project, is it possible to recover it to the original state? That's something we need to think about as well. Thirdly, it's about the long-term engineering performance degradation. As I just mentioned, on the acid condition, it does not work very well, right? So when at the natural condition, we have, for example, the acid rain sort of things. So under this condition, how long can MICP treated soil last, can keep its performance? We need a more accurate methods as well as models to quantify it. Then finally, we need to think about embodied energy, because in the MICP method, we use calcium, we use urea. So all these materials are associated with high embodied energy. So it means that regarding, it may not be a green materials for MICP. So can we use other sources for calcium, for urea, from a more energy saving and environmental friendly way? I think that's something we also need to think about. Okay, so that's my presentation today. So if you have any questions, I'm very happy to have a discussion with you. Thank you.
Thank you very much, Professor Jiang, for your very enlightening lecture. So I've learned a lot from the talk. We have seen quite a number of innovative ideas and innovative approaches developed by Professor Jiang. Professor Jiang, I'm sorry I have to go for another event, so I will ask Dr. Wang Kangda and to handle the Q&A. Yeah. Sure. Thank you very much.
Thank you.
Prof. Chu. Prof. Jiang, this is very impressive. It's very informative. So both Prof. Chu and I have some questions to ask you. The first one is for the biochar section. For the biochar section, the first one is how the biochar is introduced to the soil.
Okay, so in this test, so before we do MICP treatment, we simply premix the biochar with the calcareous sand. Then we do the MICP treatment. So it's through the premixing.
Okay. what's the size? What's the particle size of the biochar? If it is very fine, have you ever tried mixing with the biosedimentation solution and then inject into the calcareous sand?
Yeah, that's a very good question. Actually, we tried both, right? So for the particle size we use for the biochar, for the needle one, I think it's quite large. It's bigger than the sand particle size. For the powder one, I think it's like the silty soil. Silty soil. Right, but the problem is that when we mix it with water, it keeps, sink to the bottom after a certain period of time. So we don't think it's quite uniform to mix it with the solution. So that's why finally we decided to mix with the soil.
Yeah, it's because the biochar is denser than the water, so it will settle down in the water.
Yeah, with time, yeah.
So what's the source of the biochar? Is from the agricultural waste?
Yeah, it's from agricultural waste, but it's a commercial product, so we just purchase it. Yeah, but I think in the future, maybe you can make it by yourself.
Is it similar to the active carbon?
I think in some way, yes. So if we talk about the absorption ability, I think it's similar.
Yeah, because I see the ammonia can be largely reduced by introducing the biochar, right?
Yes,
if I'm not wrong,
yeah, that's true.
Good.
Yeah, but I guess maybe the active carbon is more expensive than the biochar.
Yeah, right. Another question is regarding the heavy metal section here. So you mentioned the heavy metal is treated for 7 days and 14 days. This is for one treatment, or you treat like every day? So 14 days for the 14 treatment.
Yeah, this is only one treatment. Then after that, we just cure it for a different period of time.
Okay. So what I understand is you treat the contaminated soil for one treatment only, and you left it for 7 days and 14 days and see the absorption of the heavy metal, right?
Yes, that's true.
Okay. So then the question is why the heavy metal is gradually fixed by the MICP process, even though you treat it only once at the very beginning?
Yeah, that's just I mentioned, because if we see the urea, decomposition curve here. Even if we have the contaminants inside, it still keeps going with time, but it could be very slow. So that's why we want to left it for a few days, so that the reaction keeps going.
Yeah, still on this slide, I can see the ammonia concentration for the 100 millimole is almost 0. Is this positive? Because I can see the decomposition of the urea. Some of the urea is consumed or decomposed, but we cannot see the ammonia rise up. This is because the ammonia ion is already fixed by the lead or by the heavy metals.
We guess because it's 10 days, right? 10 days is a long time, so some ammonia simply goes to the air. So when we measure it, it's just too low.
Yeah. Good. So, any question from the audience? I can see from the chat box, no one. Plus, I think that this talk is very informative. I think the audience needs some time to digest. So may I have the permission to share with audience your talk today, so I can post on the YouTube and also on the Bilibili for our audience in China?
Yes, sure, no problem.
Okay, good. So okay, I found Dr. Qianwen asked a question. He is curious using the MICP for the modeling because did not have the modules. You can go to the chat box. She have two questions. The module of the how to use MICP to minimize the rate of the urease.
Yeah, it seems. Let me see. I cannot see the chat box.
Oh, okay.
Let me see. Oh, okay. I can see.
Okay.
So thank you very much for Mintech. Because Mintech did not have the module for biological reactions, how to Mintech to mimic the rate of use? Why did you still input calcium to stabilize heavy metal?
Yeah, this is also my question. Yeah.
Okay, so that's a very good question. So yes, the Mintech does not have a biological module we can do the reaction. So that's why we control the amount of urea that has been decomposed. So we use it to represent the reaction. For example, if we only have like 10 millimole urease hydrolyzed, it means that only a little bit reaction happens, right? If we have 200, 300, it means that a lot of urea has been decomposed, right? So, so actually the graphs I show gives you the whole process, right? Assuming we can wait a very long time so that the whole process, the whole reaction could complete, right? But actually for the 100 millimole case, maybe only like 20, 40 millimole of urea is hydrolyzed. So we only see the first part, right? The first part of the graph to represent results. While for the low lead concentration case, maybe already 100, 200 millimole ureas have been decomposed, right? So we can see a lot of, so we can use, the data corresponding to 100 or 200 urease hydrolysis to see how, calcium and lead distribute at that condition. I think that's the way we try to mimic the immobilization process. Then for the second question is, you still put calcium to stabilize heavy metal. That's a good question. I don't think it's necessary, right? But because we simply use MICP, so we try to use the same MICP protocol as we did for the traditional problem. And as explained finally, right? Actually, the calcium could help to protect it by forming the outermost layer. Yeah, so that's the advantage we think we need to put calcium because it precipitates slower than the lead. So that's the reason. I hope I answer your question.
Okay, another question is from another audience. He is very curious. Can you incorporate the machine learning into MICP? Yeah, machine learning is a very hot topic in this year.
Yeah, it's a very hot topic. But for this problem, I think it's simply too complex. There's too many factors to control. Even for the chemical equilibrium simulation I explained, we simply consider the key points, not all factors, right? So I don't think at the current stage it's good to do machine learning.
Maybe in the future, maybe we can depend on the ChatGPT 5. Also some a bit more advanced techniques. I think for now, because I think MICP is still based on the experiment. And so I think machine learning only help to process the data. If you have enough data, you can use the machine learning to help you predict some trends. But for the MICP, it largely depends on the test conditions and also the soil type. This machine learning you cannot fit them in. So for the current stage, I still think we need to do much more experiment.
Yeah,
and we can gather more data. Maybe we can put it to the machine learning and to let the machine to help us predict. Oh, you put the substrates A, B, C, D, what you expect to precipitate, what's the expected soil strength, right?
Okay.
Okay, I think it's approaching 12 p.m. and it's for the lunch time. Okay, another question. Yeah, another question. From Tsinghua Ocean Engineer, one quick question for you is: can you elaborate on your opinion about scaling up? Some of the benefits.
Yeah, that's the question. I think we, as a bio-geotechnics community, we always think about it, right? How to scale up. I think, if we can solve the problems I mentioned in the last slide, right? It's sustainability for MICP. For example,
you can share your screen again.
Okay. I will show it again.
Yeah. So we can have a look.
Yeah. Okay.
Good.
So as mentioned in the last slide, so in order to upscale this technique, firstly we need to solve the problem with the MICP process itself, right? Like the treatment of the byproduct, like know better about its ecological consequences, like understand better of its long-term performance, as well as solve the embodied energy, the high embodied energy problems. That's the first thing before we can do upscale. The second thing is that we need to consider what are the best scenarios for it. For example, for liquefaction control, of course, I think MICP works very well, but can it replace traditional techniques? That's a big question. Maybe traditional one works already very well, and it's also cost effective. So it's not necessary, right, to do MICP. But in some cases, for example, for the heavy metal immobilization, we think potentially this could work, right? Because it could save money, and it is quite efficient when the contaminant concentration is not that high, right? So for this very special scenarios, probably MICP could work very well. So I think that's something we need to do. What are the directions we need to go, right? So I think that's the second thing we need to do before we can do the upscaling. And third one, finally, right, when we go to the field, we need to consider for example, the construction technique specific for MICP, right? Its associated cost, this kind of things, so that we can do field scale, right? But of course, that's always our target, right?
Yeah.
As a bio-geotechnics community, we want to do that, definitely.
Yeah. Okay. For the he also asked about the offshore foundation protection. Okay, I think what MICP can do is to protect the near sea beach. Okay, just spray it on. It can stabilize the sand. So that our group have already done some research about this one. But I can also add more about what's the benefit of doing the MICP and also upscale it. Because from the point of Singapore, because most of the cement is imported from the other nations, like in Malaysia, Indonesia, even in China. So the Singapore government also think about to produce the cement by themselves without import from the other nations, because it's a national security issue maybe in the future. So that they want to explore more about using the different method to replace the normal cement. Another is the global trend of the carbon neutralization. But for MICP, the carbon footprint should be lower than the normal Portland cement. So that's why the research is more focused on some new low carbon materials. Yeah. If you have more questions about the MICP, I think Paul Jiang is a very experienced expert in this field. So if you have more questions, you just drop an email to him, and I think his email can be found on the SU website, right? So just browse the SU website, and you have any more questions, just send an email to him, or you can also send to me or to Prof. Chu. We are all welcome. So I'd like to share my screen one more time. So we will Prof. Chu and our group are welcome all you to, tomorrow next year to Singapore to attend the 3rd International Microbial meeting conference on this special topic. So if you are interested, just click the link to register. Thank you very much. I think that's all the webinar today. Okay. Have a nice day and have a good lunch. Thank you for
Have a nice day. Thank you.
Have a nice day.
Bye bye.
Bye bye.
Bye bye.
Bye bye.